Okay, so yeah, my PhD project is on seasonal aggregations and movements of blacktip sharks at Cabo Pulmo National Park, Mexico. So shark fisheries in the Gulf of California exist because the artisanal fisheries target sharks for their meat, which is consumed locally, and for their fins, which are exported. So the management measures that are in place include marine protected areas, as well as a shark fishing ban that runs every year between May and July, and that's been in place since 2011. So my study species is the blacktip shark, Carcharinus limbatus. So this species is semi-pelagic, but it is associated with continental shelves. So it grows over 250 centimetres in total length in this part of the world, and it is near threatened on the IUCN Red List. So large aggregations of this species form in Florida, and this is quite a well-known phenomenon. And so in this photo in the bottom is a screen grab from aerial surveys conducted in Florida, where they get over 12,000 sharks close to the shoreline. But my study area is here in Cabo Pulmo National Park, which is located on the southeastern tip of the peninsula. So the protected area was established in 1995. It's a relatively small protected area of 71 kilometres squared, but within 10 years of its creation as a no-take area, fish biomass increased by over 400%. And this was because locals switched from fishing to tourism. So now Cabo Pulmo is a well-known, famous destination for scuba diving, particularly with shark species. So the NGO that I am doing my PhD with, Pelagios Kakunha, have been doing terrestrial surveys since 2016, and they noticed seasonal aggregations of sharks close to shore. So when I arrived, I decided to implement a drone project so we could use this for more accurate counting of the sharks and also identification. So my main three research questions are: how large are the aggregations close to shore in Cabo Pulmo? How resident or seasonal are blacktip sharks? And also, where do the sharks migrate after they leave Cabo Pulmo National Park? So the aerial surveys are done in a northern beach over sandy remote habitat along a 1.5 kilometre transect. So before each drone flight, I record environmental factors such as tidal height, sea surface temperature, wind speed, direction, etc. I do 2 flights, one in the morning between 9 and 10 o'clock, and another one in the afternoon between 3 and 4. It wasn't possible to do surveys between these times due to sun glare, which causes reflection to block out the transect area. So these flights were conducted weekly, and they were started in January 2019. So I'm just going to present the results from that first year, but we have continued this project as part of a long-term monitoring project. So I'm just going to show you here an example of a little bit of the transect area. So I fly the drone at a 100-metre altitude, which gives me a field of view of 150 metres. So the area that I cover is 0.255 kilometres squared. And I keep the shoreline in the side so I can accurately do the surveys every time. So to count the sharks, I extract the frames from the video, and I simply use ImageJ software. So I use the multi-point tool. So basically every time I click the screen where there's a shark, the software tallies the number of times I've clicked the screen. So our results, so we counted between 1 and 1,086 sharks along the transect area, which gave us a maximum density of 4,826 sharks per kilometre squared. And this data was published last year. So we used a generalized additive model to basically model the environmental factors with the abundance. So we used a negative binomial distribution with a backwards stepwise method to determine the final model. So each variable was removed and the model with the lowest AIC score was used. So my final model included the factors sea surface temperature, wind speed, photo period, and time of day. So here are the results. So what we found is that there were significantly more sharks in the afternoon than in the morning, and this coincided with a peak in water temperature. which means that we think that the sharks are close to the shore, potentially for thermoregulation, coming closer to the shore in the late afternoon. We also think that this could be for refuging against predators. So, for example, in Florida, the great hammerhead sharks feed on the blacktip sharks, and we do have orcas and larger shark species in the area. We also found that sharks were absent from the shore when water temperature was higher than 25 degrees. And we also found that wind speed was a predictor of shark abundance. So we flew the drone up to 15 knots. Any higher than that, we decided it wasn't a good idea because there was too much water movement that could mask any sharks present. But we found that, the calmer the water was, the higher abundance of sharks that there were. And this, again, could be because of less disturbance, for this to be like an area where they rest before they move offshore to feed. So we also get a lot of bait fish in winter, which could be also another reason for these huge aggregations. But interestingly, a lot of the time when we see these big schools of bait fish, the sharks were not feeding or hunting. They were simply slowly swimming through these schools. There was only one predation that I observed, which I'm going to show you now. So basically the sharks, it's hard to see, but it's chasing a very small sardine in the wave break. And I think that this is definitely more common than my drone surveys show, because a lot of the locals would send me videos of this happening, but usually later on in the day after I've completed my final drone survey. So that's something I could do in the future, is include a later drone survey to see if I can document this predation more often. So yeah, for the seasonality, the sharks were present between December and April. There was a few sharks in May, but much significantly less. And so they're absent from the shoreline in the summer months. So we also used acoustic and satellite tagging. So we managed to tag 27 sharks internally with acoustic V16 transmitters. We tagged 17 females and 9 males, and we also tagged 8 sharks with satellite spot toad tags from wildlife computers, and we tagged 5 females and 3 males, and the average size was 210 centimeters. So all these sharks were adults. So for the acoustic telemetry, we have an acoustic array within the park along the reef area. This is a abacus plot. So we have the names of the receiver locations here. And then we've been monitoring these sharks since 2015. And again, like with the drone surveys, we can see that the sharks are absent. These gaps in the detections are in the summer. So that's why it was important to use satellite tagging to see where the sharks go after Cabo Pulmo. So we got this really cool track from a female shark that we tagged back in 2019. So it traveled over 900 kilometers all the way up the Gulf of California, which was pretty cool. So the sharks are probably following like cooler, more productive waters. Like here in the middle is the Mid-Drift Islands, where there's a lot of upwelling and productivity. We also tagged another shark that made its way to Loreto within a month, again, a northerly migration. Unfortunately, we couldn't tag in 2020 because of COVID-19, but we managed to tag some sharks last year. And we have one shark that traveled in April to May up here. And finally, a male that made like a northeasterly migration in May. So another reason for this migration could be the females migrating to nursery areas. So these are 4 like locations that have been described as potential blacktip nursery areas that needs further investigating. But we had a unfortunately dead female blacktip on the survey beach back in 2013. And we cut her open and there was 8 pups inside, and the pups were just below birth size. So basically the females could be using the shallow shoreline, waters of the shoreline, to speed up gestation before they make their northerly migration to nursery areas. So the next step in the research, which will be for my postdoc, will be to find out if the aggregations are male or female. We've already attempted to place block cams in the aggregation site, but the sharks seem to be completely deterred by the cameras. So I need to find a way to be able to see if the aggregations are male male or female, because that would be really interesting to find out. Unfortunately, a lot of the sharks are caught. So 7 of the 27 sharks that we tagged were never detected again. And this image here is from inside Cabo Pulmo National Park. So this is a simplera. It's basically a buoy with a hook. And this was placed outside of the park, but due to like bad weather, it drifted inside of the park. So there's a lot of pressure on these sharks from the fisheries. And also all of the 8 spot tags, only 4 transmitted. And all of the 4 that did eventually transmitted from land within a few months, which indicates that they were caught in a fishery. So the sharks are protected between December and April when they're within the protected boundaries. But this research shows that once they leave these boundaries, they are at risk to fisheries. And the shark fishing ban runs between May and July. So unfortunately, the sharks are migrating just before the fishing ban. So this image was actually taken just last week in a fish market in La Paz, because the sharks this year have migrated slightly earlier than usual, and they're already being caught by the artisanal fisheries. So unfortunately, the fishing ban doesn't quite cover their migration. So to conclude, how large were the aggregations? So over 1,000 sharks at times, more in the afternoon with a peak in sea surface temperature and at low wind speeds. How resident or seasonal are black-tip sharks? They're only present between December and April. And after they are in Cabo Pulmo, they seem to do a northerly migration. But obviously we need to tag more sharks with satellite tags to see if they leave the Gulf of California, or to basically highlight any more important areas that these sharks go to, particularly the seamounts, Las Animas and El Bajo, where we have sharks detected as well. So I'd like to thank all the donors and Pelagios Cacunha and my university, and also, thanks for listening.
Great, thanks, Catherine.
Thank you.
We'll have our next speaker be Paul, apologize if I get your name wrong, Preciado Gonzalez. He'll be speaking on movements of Manta birostris in the Mexican Pacific.
Hi.
Hi.
Katie, could you stop your sharing screen, please?
Catherine? Hello?
Katie? Oh, yeah.
Okay, there we go. Okay.
Okay. Okay, well, good afternoon, everyone. Thanks for introducing me. Today I'm going to talk about a little brief summary of my results of my master project. Well, the oceanic manta mobila virostris is the largest ray in the world. This inhabits in shore and offshore areas of tropical and temperate waters around the world, related mainly to upwelling areas, oceanic islands, and seamounts. This is considered a solitary species with the ability to travel long distance between aggregations areas searching for food. But these long distance movements occur at different spatiotemporal scales, where mantas could be susceptible to natural or anthropogenic injuries during these movements. The target fisheries and bycatch are the main activities that led the giant manta race to be endangered by reducing populations around the world. Therefore, understanding its movement pattern is critical for implementing effective management strategies for their conservation. Advances in sensor attached to animals have led to remote study of marine species movements, behavior, and habitat use. The acoustic telemetry is a technique that requires the use of acoustic receivers to record the signals emitted by an acoustic transmitter placed on a marine animal. However, these issues are not currently well understood for the oceanic manta ray mobila virostris. For this reason, this project aims to determine the movements of mobila virostris in the Mexican Pacific using passive acoustic telemetry. We focus on 3 known and separated manta aggregations areas in the central northern Pacific, such as Revillagigedo National Park, Cerralbo Island, and Banderas Bay. Just to be a little bit more specific, the Revillagigedo National Park is an archipelago of 4 different islands, such as Clarion, Socorro, San Benedicto, and Roca Partida. For this project, we use acoustic transmitters and acoustic network deployed in the Mexican Pacific at different places around Baja in the Pacific side and inside the Gulf of California, as well in the archipelago Revillagigedo National Park, and close to Banderas Bay and in Clipperton Atoll. Okay. The acoustic network is made up of an acoustic station consisting of a receiver like this in the picture, attached to a mooring line to a steel chain affixed to the bottom and suspended in the water by a buoy. Each acoustic station is your reference, and with it was possible to calculate the straight line distance between the acoustics receivers to estimate the distance between the receivers in the network. Okay, about the results, from 2012 to 2018, 61 manta rays were tagged in the Mexican Pacific. 27 were tagged in the archipelago of Revillagigedo, 31 were tagged in Banderas Bay, and the last 3 manta rays were tagged around Cerralbo Islands. Okay, we analyzed the residence of the manta rays at the 3 different regions, and we found a same pattern of the manta rays. The manta rays remain at the site during daylight hours while moving away at night. The pattern was to 6 in the morning to 6 in the afternoon. As for the results at the local movements, we found that mantas have preference for moving in the southern part of the Banderas Bay, mainly at Los Arcos and Yelapa. Okay, but in the Revillagigedo National Park, we also found movements around the islands, but we also find movements of the manta rays between the islands, specifically more between Socorro Island and San Benedicto Island. The thicker line means a greater number of movements of the manta rays in the area. And we found that a corridor, a preferred route of the manta rays is between El Boiler and Punta Tosca. 2 manta rays move to Roca Partida from El Boiler and Punta Tosca as well. However, we also found long distance movements where the 5 manta rays moved just between Banderas Bay and the Revillagigedo National Park. Then, based on the movements that manta rays made, we calculate the total minimum distance traveled for a manta rays. The first manta ray was tagged at Banderas Bay, close to Canilla, and the manta traveled to Roca Partida with a straight line distance record for the manta rays in the Mexican Pacific is about 698 kilometers. And then the manta rays moved to Socorro Islands, specifically to Punta Tosca, then Cabo Pierce. Then the manta ray moved to the San Benedicto Island, to the canyon. And after that, the manta was recorded in Yelapa inside the Banderas Bay. But 3 months after, the manta rays move again to Socorro Island, stay around Socorro Island, move to the San Benedicto Islands, between the canyon and the boiler, and then was recorded again inside Bahia Banderas. This manta was a male, and the total straight line distance calculated, it was around 2,735 kilometers. Another one was a female that was detected and tagged and detected for the first time in Bahia Banderas and moved to El Boiler at San Benedicto Islands. The total straight distance was around 60 kilometers. Okay, another one was a manta tagged at the Revillagigedo National Park, and this manta race with an unknown sex was moving around San Benedicto Island, Socorro Islands, and then was detected inside of Banderas Bay. The next one was a male target at close to Los Arcos in Bahía de Banderas, and then moved to Socorro Island and was swimming around the main two spots in the picture. We don't know how this manta was moving, but we only have detection in both sides. But then the manta rays moved to Bahía de Banderas because it was detected at Chime. The total straight line distance was around 2,239 kilometers. And the last one was a male that target Banderas Bay and moved to Punta Tosca in Socorro Island. Conclusion so far, the movements of oceanic manta rays were recorded in the Mexican Pacific for the first time, and the analysis demonstrate connectivity into two regions in the Mexican Pacific Ocean. I just want to say thanks to all the organization and institute to help me, and thanks for you for attending my presentation.
Great. Thanks very much, Paul. Our next speaker, Elaine Rodriguez, is not here, so we're moving on to the next speaker, Francisco Cordova Zavalada. Will be speaking on the artisanal fishery of elazobranchs in North and Central Peru, gill nets as a case study. And I apologize, Francisco, if I pronounced your surname incorrectly.
Hello, never mind. Well, I know that this is a Northeast Symposium, but what if we move to the southern region, more specifically to Peru? So Peru is located in the transition between tropical and warm temperate waters. So it means that there is a high biodiversity of species, including 68 shark species and 43 batois. As most of you know, Peru is a fishing country, so these species are not the exception. So fishery of the elasmobranch is totally artisanal, and the fishing gears used is according to the target species. I mean, long lines are used for targeting blue sharks, mako sharks, while gill nets are used to pursue some kind of hammerheads, treasure sharks, and also some eagle rays. The relevance of the activity is the trade of the fins, which go to the Asiatic market, while the meat is consumed locally under the denomination of toyo. So San Jose and Salaberry are the main landing points in northern and central Peru, respectively. So they represent key localities for fishing monitoring. So our study area was focused on this particular region. San Jose in the Lambayeque region and Salaberry in the La Libertad region. So for this, we used an onboard monitoring program between 2015 and 2019, gathering some information about fisheries. For this, we tried to characterize the fleet. First, all about the dimension of the fleet, also the material of the hull, refrigeration system. We also wanted to expand the knowledge about the fishing operations. I mean, all about the nets they use, the type, the material, the mesh size they use, target species, some days at sea, and also to complement information, some economical aspects as cost and benefits. To calculate fishing effort, we multiplied the length and the height of the net by the soak time, I mean the time the net was deployed at the sea. And to calculate the capture per unit of effort, we divided the number of individuals between the fishing effort. And it's important to mention that all of this was by set. To gather some information about biometric, we make some measurements about batoids and sharks. And for the results, we got information from 11 vessels from Salaberry and 10 from San Jose. Which is important here to see is that fleet from Salaberry was bigger than Salaberry, but this is not, we cannot consider that because they are bigger, it is a major fishery. Then about the whole material, all of the vessels were made of wood, and the refrigeration system was in most of the cases was isothermal hull plus flake ice. Regarding fishing operations, we saw that Salaberry and San Jose share drift nets as main net types, but also Salaberry used mid-water nets to capture some kind of eagle rays, while San Jose bottom nets to capture the Mustelus sharks. In the case of the material, Salaberry and San Jose use multifilament nets, while San Jose use monofilaments. And it's important to see here, while we compare with mesh size, San Jose, the lowest mesh sizes for about 4 to 6 inches, were monofilaments and also deploying the bottom to capture Mustelus. The days at sea, well, in Salaberry, they spend more days at sea to capture because they use far away areas in comparison to San Jose. And as I was saying, Salaberry had the target species of smooth hammerhead, while in the case of Batois, the eagle rays. In the case of San Jose was the humpback smoothhound, the main shark species, while for Batois, also the eagle rays, the Miliobatis in the case of Peruvianus and Chilensis. Some general information about this study, more than 500 onboard trips, more than 3,000 sets analyzed, almost 142,000 elasmobranch captured, comprising 39 species of elasmobranchs, which were 22 sharks and 17 batoids. And in the case of number of sets, the Salaberry made more sets per trip than in the case of San Jose. Here's a picture of some of the captures we recorded. We can see mobulids, some treasures, mustelus, blue sharks, eagle rays, guitarfish, angel sharks. There is plenty of species here. Regarding the fishing effort, it is important to see that the Salaberry has the most important fishing effort in comparison with San Jose. But if we see the capture per unit of effort, we can see that San Jose has a major capture per unit effort in comparison with Salaberry. This can be considered good because with low fishing effort, you can get more individuals. But it is important to mention that only we are considering individuals, not the weight of the individuals. So you are going to see this when we see the economical aspects. About the sex and size analysis, here's the histogram of the most important sharks captured. And if we consider the first size at maturity, we can see that there is 6 species captured when they are juveniles and only 3 when they're adults. In the case of batoids, if we consider also the first size at maturity, the whole species are captured when they are juveniles. So this is something important to take care about if we want to get this activity sustainable in the following years. What I was saying is about the cost of fishing trip. The cost in Salaberry is much larger than in the case of San Jose. And if we can see the benefits in San Jose are much larger than in the case of San Jose. There is also some exceptional fishing trips in which we can consider there is a boom of an abundance of sharks and batoids. But it's important if you can see that there's also some fishing trips in which the benefits are zero. I mean, they only get to pay what they invested. And also you can see that there are some trips in which they return with debt. So there is a question here: is this a profitable activity? And finally, as I was saying, here look that Salaberry has more benefits than San Jose, but the capture per unit of effort was higher in the case of San Jose. So maybe we can explore or to complement this study by considering a mean body weight of each species to compensate this difference. No, maybe for a later study we can see that and complement the study. For here, thank you. I'm willing to take some questions at the end of the session.
Great. Thanks very much, Francisco. We'll move on.
We will to our next speaker, who's Felipe Galván, to give us an update on ongoing research on sharks in the Gulf of California.
Felipe, we can see your screen, but the PowerPoint hasn't started yet.
Okay, do you see my screen?
We see your screen, yeah, but you just the
perfect.
PowerPoint
Okay,
hasn't
sorry.
started. Yeah, that's okay.
Sorry. Okay.
There we go.
Okay, got it. Okay.
Perfect.
Okay, I'm going to talk about our research in the Gulf of California, studies on sharks and rays. Well, Carla Garces explained about the oceanography, and then it's a very interesting area. As you can see, the water in these areas is temperate waters, hot waters. There are deep waters in the area. In the eastern coast is a moody area. There are mangroves area, very important for nursery areas for different shark species. But in the western coast is more rocky areas with islands, etc. And then it's a very, very important ecosystem for sharks. But the problem is sometimes the human with fisheries affect this ecosystem. In the year 2000, no more sharks in the area for almost 10 years. We don't know exactly what happened. Probably the effect on the temperature of waters, but we think more on the effect in fisheries, because before the year 2000, fisheries came here in this area, and then they put nets and longliners and fishing for more than 10 years. And then in the year 2000, no more sharks. But we have now sharks. And that is the reason that, again, we want to do research to try to know different information on biology. And then this is the area mostly from the Santa Rosalia to La Paz, where we are doing different research on sharks. We have now a record using our data of 19 sharks and 22 ray species. Okay, the most important sharks is the Pacific sharp-nosed shark and the Sternalunai and the angel sharks and pelagic treasure, spotted hammerhead sharks, and some smooth hound, the brown smooth hound, and other species. But these are the most important in fisheries, mostly in artisanal fisheries. For rays, the catches are more for the guitar. Two species of guitar fish is the this species, Pseudobatus glaucostigmus and Pseudobatus productus, or booty. We need to do more research to recognize what species is because some information mentioned that probably is Pseudobatus booty, and also the butterfly ray and the golden cownose ray. And then these species are very important in the fisheries, and then the humans are feeding on these animals. And then it's very important to do biological studies to know reproduction, the age, growth, feeding habits, to know the demography. All this information is important to give data for the protection or management of the different species in the Gulf of California. And then also we're doing the trophic ecology. Well, some results are shown in this symposium using the stable isotopes and fatty acids. And also we are doing the research on the contaminants, metals, and some pesticides. Because it's also in the area, especially in Santa Rosalia area, there are mines, and then mines probably they produce some contaminants and affect the meat, the rays of sharks' meat. And then that's the reason that it's important to do all this research on this area. Okay, and then the most important is our students and researchers in our projects. In this project from Cicimar, we have a tons of experience working with us, and thanks for your help doing this research for this area in Mexico. Okay, thank you.
Thank you very much, Felipe. Short and sweet talk.
Okay, thank you. Okay, our last session of this symposium with Ana Maria Moncada Rosas, with the talk about the feeding habits of the brown smooth-hound Mustelus henley on the western coast of Baja California Sur, Mexico. Ana Maria?
Yes.
Are you there?
Yes. You're able to see, right? You're seeing my screen, right?
Yes.
Yes.
All right, should I begin now?
Yes.
Okay, good afternoon. My name is Ana Maria. I'm going to be presenting my project called Feeding Habits of the brown smoothhound called Mustelus henley in the western coast of Baja California. Okay, first of all, we have that this species is distributed in the western coast of the Pacific Ocean, from North California to Colima and from Costa Rica to Peru. This species is one of the most dominantly caught in the artisanal fishery.
Ana, ¿puedes ponerte más cerca del micrófono para escuchar? No es muy claro el sonido.
Sí, claro. Ahí ya está mejor.
Ok, perfecto.
Listo. This species is one of the most dominantly caught in artisanal fisheries, and it's of great ecological and economical importance. Previous studies have reported its diet to be composed of mainly fish, cephalopods, and crustaceans, but this information must be updated. Okay, and knowing the trophodynamic of these species is highly relevant, for which this study aims to determine the feeding habitats of Mustelus henley. And for this, we need to identify the main prey items in the diet, estimate trophic spectrum width, feeding strategy, and trophic level, and determine diet changes among sex, sexual maturity, and sampling years. Shark specimens were obtained from commercial fishing gill nets in artisanal fisheries from the 3 points you can see on screen. Sampling took place between 2014 and 2018, excluding 2017, and it was carried out through August and September. We registered the sex and total length of every shark, and specimen and stomachs were removed and preserved for their further analysis. Stomachs were defrosted, and the degree of stomach fullness was determined according to steel rod and color and assigned to one of the 5 categories shown in screen. During stomach content analysis, prey were classified into different taxonomic groups, washed and weighed. Preys were assigned to one of the 4 degrees of digestion, and then they were identified to the lowest taxonomic level possible. To determine whether the number of stomachs analyzed was adequate to characterize the trophic spectrum, cumulative prey curves and variation coefficient were calculated, where CV values of under 0.05 showed an adequate representation. The most important prey species were within the trophic spectrum were identified using numerical, gravimetric, and frequency of occurrence method. And once these values were obtained, we calculated the index of relative importance, which allowed us to make comparisons to previous studies and the prey-specific index of relative importance. Trophic niche breadth was estimated using Levi's index, which showed us if they were specialistic or generalistic predators, and this was complemented with the Amundsen's graphical method. The trophic position was estimated by means of the equation invented by Kristen and Polly, and a Permanow analysis allowed us to identify statistical difference variations amongst the diets. If they were present, we used a simpler analysis to show the preys that mostly contributed to these differences. As for results, a total of 150 stomachs were analyzed for this study, and samples were representative to describe general diet and make comparison between sexes, maturity states, and all sampling years except for those of 2014. These stomachs were included in the diet description of the general topic. 82% of the stomachs belonged to females, and most of the individuals captured were mature. Only 7% of stomachs were empty, and over 50% of those who had contact had a fullness ranging between 51 and 75%, which could be related to the slow digestion of most of its prey and predominantly nocturnal feeding habits. The general trophic spectrum of Mustelus henley was composed of 43 preys, of which crustaceans were the most representative according to the prey-specific index of relative importance. Hemischila californiensis and Pleurocondes planipes represented over 48% of the diet, and this might be due to 2 reasons: their high abundance, permanence throughout the entire year, resulting in these organisms being very easily accessed by the predators, and 2, their very low caloric input, which makes it necessary for the predators to eat various of these individuals. Bony fish were the second most important prey, and cephalopods had a very small contribution to the diet, contrasting to what was reported for this same shark species in Punta Lobos, which is located in the southernmost portion of the western coast of Baja California. The stomach content analysis showed 2 feeding habitats for Mustelus henley, as it consumes both pelagic and benthic prey. Bony fish have a higher importance in the diet of these sharks than any other of the same genre, of the genre Mustelus, and this is mainly due to the fact that they have paved teeth with tall cusps and accessory denticles that allow them to catch and hold on to fish very efficiently. A low value of the Levine's index was found, which classified Mustelus henley as a specialistic predator, coinciding with what's found by multiple authors. However, the specialist classification may be a consequence of the high availability of Hemischila californiensis and Pleurocondes planipes in the sampling area, as a large number of individuals fed on them in high proportions. Therefore, it could be established that Mustelus henley is more of an opportunistic predator, even selective opportunistic. And the graphic analysis showed exactly this, that it has a high specialization between organisms, indicating a high contribution between individuals towards the width of the niche. Significant differences were found between the diets of males and females of Mustelus henley. However, these differences rely mainly on the proportion of prey and composition of secondary prey. They could not be the result of a sexual segregation on feeding, but rather than a greater use of habitats from behalf of the females. And this has been previously reported by various authors in the Gulf of Uyuela and Pacific coast of Costa Rica. Two crustaceans represented more than half of the diet in females, these being Hemiscylla californiensis and Pleuroconda planipes. And for these species, many fish, especially Poristes and Prionotus, were registered as secondary prey. On the other hand, Hemiscylla californiensis, Sardinopsagas, and Pleuroconda planipes were the most important prey in male diets, whereas Delphinidae, Prionotus, and Lepophidium were secondary prey. According to the Levine's index, females are slightly more selective about prey than males, while still having a predominant opportunistic feeding strategy, where different individuals specialize over different prey. Mustelus and Ley presented differences in the diet between mature and immature organisms, in agreement to what was reported in previous studies. Presenting changes in the diet as individuals increase in size or sexual maturity is a widely reported strategy in elasmo ranks in order to reduce intraspecific competition. Prey size may be an important factor related to the different feedings of immature and immature organisms, but for this species, apparently a spatial segregation related to the maturity stage might be the correct scenario. So although crustaceans were important in both of the maturity states, they represented 78% of the diet in immature organisms. And this, together with the presence of bony fish with benthic habit in their diet, may indicate a wider explanation of this habitat from their behalf and a wider exploration of the water column from the behalf of mature organisms. The Levi's index classified mature organisms as specialists. However, after analyzing this value with the Amundsen graphic method, it can be established that behavior is more opportunistic and that organisms consumed with more frequency could have influenced the low value of the index. Okay, so differences were also observed in the diet of Mustelus and Ley between 2015, 2016, and 2018, since although the most important preys were the same throughout the 3 years, the percentages of contribution to the diet and the diversity of secondary preys varied remarkably. These differences may be related to variations in the environmental conditions present during these years, since these have a direct effect on the availability and diversity of food in the area. Between 2013 and 2016, the Pacific Ocean experienced atypical weather patterns, which caused an increase in about 4 degrees Celsius on the surface of the temperature, affecting oceanic processes, including declines in the primary productivity, changes in abundance, composition, and diversity of many of marine species. Especially during 2015 and 2016, warm water species increased their distribution, which might explain why there is such a high incidence of prey like Pleuroconda planipes during the diet registered in 2016. Levi's index also suggested once again the specialist strategy, which could be taken as a selective opportunist towards crustaceans, because those are the most abundant prey in the habitat that they are, and also the big plasticity and accommodation of the shark to the food that's available around them could be an indicator of its great adaptability and plasticity, for which they might be very resilient. Okay, and for last, I would like to thank you, Dr. Felipe Galván, for directing this project, Dr. Arturo Tripp and Mariana for their guidance, to thank the Instituto Politécnico Nacional, Cicimar, Proyecto TIburones&Rayas, and University del Bosque for all the support throughout this project. Thank you very much.
Okay, Ana, thank you. Okay, the next speaker is Ángel Moreno with the talk about the organochlorine pesticides in fish shark species from the western coast of the Gulf of California, levels, species-specific accumulation, and health risk assessment.
Hello everybody. Thank you, Doc. So my name is Ángela. I'm a PhD student in Cicimar IPM, and today I'm going to talk about organochlorine pesticides in fish shark species from the Gulf of California. Organochlorine pesticides are organic chemicals that have been used in numerous and diverse applications since the 30s. Despite their great applicability, mainly in agriculture and for the control of vector disease, due to their high toxicity, on wild animals and humans, they were included in 2001 in the group of persistent organic pollutants, and their use and manufacture have been banned almost everywhere in the world. However, due to their high environmental persistence, their ability to disperse, their reaccumulation power, and their continued use in some countries, they are still present everywhere in the world. Organochlorine pesticides are absorbed by marine organisms, mainly from the diet. Most of these chemicals are lipophilic, very lipophilic, and difficult to excrete, so they tend to accumulate in the fatty tissue of organisms. Therefore, when the exposition is higher than the elimination rate of the organism, organochlorines bioaccumulates. That means their concentrations increase with the growth of the animal. However, the bioaccumulation is a process influenced by numerous physicochemical, ecological, and biological factors, which imply that there can exist large inter- and interspecific differences in the accumulation. In addition, these contaminants tend to biomagnified. That means they increase in concentrations with each level of the food web. So marine apex predators such as many sharks tend to accumulate higher concentrations than lower trophic levels. This makes sharks potentially more vulnerable to their toxic effects, and because many shark species are exploited in fisheries for consumption, they are vectors of contaminants to humans. Therefore, and due to the potential toxicity of organochlorine pesticides to sharks and humans, the identification of species and population groups of sharks prone to the exposure and accumulation of pollutants facilitates the focus of risk assessment and management efforts. Although the Gulf of California is considered a relatively pristine environment, its fauna has been exposed to organochlorine pesticides, given that the continental coast of the Gulf of California represents more than 30% of the area designated for agriculture in Mexico. However, until our work, there was no information regarding the levels of organic pollutants in predator elasmobranch species of fishery importance. Therefore, our study species are the three most exploited sharks in this area: the scallop hammerhead, Sphygmolewini, the Pacific shark-nose shark, Chrysopyranodon longurio, and the Pacific angel shark, Squatina californica. We took samples of liver and muscle of twin specimens of each species in 2 fishing camps from the western coast of the Gulf of California. The lipid-rich liver of sharks is the main accumulation organ and reflects the maximum load of organochlorine pesticides that can eventually cause toxicity when they are distributed to other tissues. The muscle samples represents the edible tissue of sharks and allows to evaluate the risk associated with organochlorines due to the human consumption. The determination of lipid content and the identification and quantification of pesticides was performed in the University of Baja California using last generation equipment and following the required quality controls. For the data analysis, we used non-parametric tests to identify differences and trends between groups of organisms. 23 of the 29 pesticides analyzed belonging to different groups were identified in the liver. The DDTs group, which were widely used in agriculture and for malaria control, had the highest concentrations. We also found chlordans used for termite treatment and Mirex or chlorobenzenes, which had mainly industrial applications. Other legacy pesticides were identified at lower levels. The liver of the sharks analyzed in this study contained low levels of DDTs compared to concentrations reported in shark species around the world. Due to the magnitude of the differences, our results seem to reflect lower historical inputs of DDTs in the Gulf of California compared to other regions. Even reducing the influence of lipid content in organochlorine concentrations by lipid normalization, the angel sharks had significantly low levels. Since the 3 shark species are tertiary consumers in the Gulf of California, one possible explanation of the low organochlorine levels in angel sharks is that this species experienced a lower exposition due to its spatial ecology. We found that the three species also show different lifetime accumulation trends, confirming that multiple factors influence the accumulation patterns in sharks. In the case of hammerheads, a moderate relationship was observed between the total length of the juveniles and the concentration increase of organochlorine pesticides. This suggests that the elimination rate of these compounds is lower than the exposure, resulting in their accumulation over time. Considering that the hammerheads analyzed in this study were immature and the relative long lifespan of this species, Sphyrna lewini seems to have the highest accumulation potential. The concentration of pesticides detected in one of the two neonates analyzed was above the threshold value for adverse effects in fish in early life stage. Since organisms in their early life stage are particularly vulnerable to organochlorine's adverse effects, and due to the poor conservation status of Sphyrna lewini, it would be advisable to carry out more studies in this population group. In the case of shark-nosed shark, they show a U-shaped lifetime accumulation curve, as it is called for describe a period of growth dilution that is observed in juveniles and a period of increasing concentrations after maturity. We also see that the increase in concentrations is higher in adult males than in adult females, possibly as a result of maternal offloading of pesticides. In the case of angel sharks, there was not a relationship between the size of the individuals and the organochlorines concentrations, suggesting that the uptake and elimination rate are similar. In mussel, the number of organochlorines identified and their concentrations were lower than those in liver, which is associated with the low lipid content of this tissue. These concentrations were between 3 and 5 orders of magnitude lower than the limits established by different agencies for human consumption. Therefore, the consumption of these sharks caught in the study area does not imply risks to human health associated with organochlorine pesticides. However, since all three species are also exposed to other toxic pollutants in the Gulf of California, the evaluation of human health risk posed by multiple contaminants associated with consumption of this species will be advisable. So the main conclusions of this study are three. Sharks from the Gulf of California are exposed to a wide variety of organochlorine pesticides, although their concentrations are relatively low. If contaminant concentration is correlated with its effects in organisms, hammerhead may have more probabilities of negative impacts, and angel shark lower probabilities of toxic effects. The consumption of this species does not seem to imply risk to human health associated with organochlorine pesticides. And if somebody is interested on this topic, it is already available, the first publication about it. And if you have any question, this is my email address. Thank you very much.
Okay, thank you, Angela. Okay, the next talk with is a pre-recorded talk is Ariadna Lara, mercury and selenium in the meat of the smooth hammerhead shark, Sphyrna saccina, suitable for human consumption. Ariadna, are you there?
Yes, but I record my presentation, and could you put it on? That's okay.
Oh,
okay.
Okay.
Thank you.
You can be the talk. Okay.
The title of our work is Mercury and Selenium in the Middle of the Smooth Hammerhead Shark, Sphyrna saccina, suitable for human consumption. Trace elements are classified as essential and not essential, depending on the roles in the organisms. Essential elements are required for the metabolic and homeostatic functioning in the organisms. They can cause damage if they are at elevated concentration while not essential element, while not a specific biological function except toxic effects even at minor concentration. Essential and non-essential elements found in the environment are solved from diversity natural and anthropogenic processes. Recently, coast and marine ecosystems are becoming repository sites of heavy metal due to urbanization and industrialization processes worldwide. The bioaccumulative process persists, and toxicity of trace elements often results in negative impacts in the marine ecosystem, mainly the high trophic level species. Mercury is viewed of the element most studied for the potential damage it can cause to organisms. It most investigated that evaluate the concentration of mercury. They also measure the concentration of selenium because it is an essential micronutrient for organisms and has an antagonistic effect with mercury, inhibit its toxicity and accumulation in organisms. Sharks are highly vulnerable to contaminant accumulation during their lifetime due to their longevity, low metabolic rate, high trophic position. Evidence of toxicity in high of trace metals in sharks poses several threats of the marine environment, including human. The Mexican shark fisheries ranks among the most important globally. Juvenile smooth hammer shark, Spina sicena, are one of the most cautious species of the Pacific coasts of Mexico and the United States. An increase in human damage for shark products has receded worldwide, giving as a result the necessity of examining trace element level in shark tissues for better understanding of the potential impact of human health. Today, numerous studies have focused on element accumulation in adult sharks of different species. However, juvenile sharks are the foremost important bioaccumulation study because they commonly reside in coast areas that are greatly exposed to higher concentration of anthropogenic populations. Due to this reason, the objective of this investigation was to determine the concentrations of mercury and selenium in the muscle of Spina sicena and to assess the risks to human health due to its consumption. The sample were taken in fishing camp of Bahia Tortugas, located on the western coast of Baja California Sur. Fishing is the most important economic activity in the region. For the all organism of this species, Spina sicena, the total length of these organisms was measured, and they were classified into male and female. We taken a fragment of muscle to proceed to laboratory analysis to determine the concentration of trace element, mercury and selenium in this case. This graphic shows the concentration of selenium and mercury for female and male organisms of this species. In this case, the average of concentration of selenium and the average of concentration of mercury, and the mean concentration of mercury in the muscle in this study was relatively lower than the value reported in juvenile of this species or the other plates, for example, González Pesana, Peru, and Musgrave in Baja California Sur too, and is lower this concentration than the maximum limit established by the Mexican norm for human consumption for mercury. And that is suitable for human consumption. This species, these organisms that this study, the following table shows the percentage of weekly mercury intake compared to the weekly provisional tolerance intake. The weekly tolerance intake is defined as an amount of a toxic substance that can be consumed without presenting any risk to human health. In this case, in the case of children, present the highest weekly potential tolerance for mercury. This is 2.06% being able to ingest 0.3 kilograms of spirulina meat. However, the man, in this case, the man weight can consume a maximum of 2.6 kilograms per week of meat for this shark. The molar ratio mercury selenium was 1 to 1.3. Molar excess indicate that more than one molecule of selenium is present in the tissue and could be involved in the formation of mercury selenium complex for the effective detoxification of mercury toxicity. And the calculated selenium health benefits value was negative, but it was not significant because of a similar concentration of mercury and selenium. For the conclusion, the consumption of meat of filnacigena does not represent a risk associated with mercury intake because the average concentration is below the maximum permissible limit for human consumption. Considering the weekly intake, the level of risk of toxicity for mercury is low. However, it should be noted that a specific sector of the population, such as fishing community, included more fish in the diet, so the level of risk is difficult to accept based on individual species. More details of the rate of ranking stakes in this community are needed to properly assess the level of risk. Thank you for your attention.
Thank you, Ariadna. Okay, then the next speaker is Joshua Bowman, White Sharks, Carcharodon carcarias in Oregon, expanded niche or undescribed habitats.